Hello and welcome to my YouTube channel, AnatomyMentor. Today, I'll be discussing a topic of histology, that is the histology of the cell membrane, also known as the plasma membrane. So this is a very important topic, and let's get into it. What are our learning objectives for today? Well, we shall discuss the cell, identify its method of studying. We shall talk about the structure and function of the cytoplasmic organelle, which is the cell membrane. You must have noticed I call the cell membrane a cytoplasmic organelle. Now, why is this known as an organelle? This we should also discuss in the upcoming slides. So, what is a cell? Now, we all know that the cell is the unit of life. This is what we have been taught in our textbooks, in biology classes. It is the basic unit of structure and function of all living organisms. But keep this in mind. This is not a simple unit. This is a very complex unit and a highly developed unit. What are the examples of cells around us? Well, if you look over here, you can see there are some unicellular organisms like the amoeba. Plus, we know that there are cells present within plants and also within animals, specialized cells like red blood cells or nerve cells. And the bacteria, they are also cells. When was the cell discovered? Well, if we recall, Robert Hooke in 1665. You must have read this name in your textbooks. If you recall from your biology classes, you must have been familiar with this name. Robert Hooke was a scientist who discovered the cell. He observed the cell under the microscope, and this was actually a plant cell. It was the cork cell walls, which he saw. And he named it the cell, which is derived from a Latin word, cellula, which literally means a small room. And if we go into the historical aspect, there were many theories proposed by different scientists about cells. Here is a scientist who was Scheiden, and he proposed that all plants are made up of cells. And there was the scientist Schwann, who proposed that all animals are also made up of cells. And Virchow was the scientist who proposed that cells, which are already existing cells, give rise to new cells. Now, since we're talking about cells, I want you to understand an important concept of the surface area to volume ratio. Now, what happens is that as a cell grows in size, its surface area to volume ratio decreases, which means that it will have relatively less surface area compared to its volume. And as this happens, it makes the exchange of materials more difficult because of less availability of the surface area, that is the cell membrane or the outer boundary of the cell. So it is necessary that as the cell grows, as it gets larger, it should also undergo a division, a mitotic cell division, otherwise it will no longer be able to survive. So this is why cells divide, and this is why cells are small. So here you can see if this box is representing a single cell, we can see the boundary is the cell membrane or the surface area of that cell. Now, if it divides, it will give rise to many cells, which will be smaller in size initially. And as you can see, the surface area now, that is the membrane available for the transport of substances, it has greatly increased in relation to the volume. Now, how do we study cells? There are different instruments available for helping us to study cells because our human eye is not capable of visualizing a single cell. This is because the resolving power of the human eye is limited to 0.2 millimeters. Now, what is the resolving power? It is the ability to differentiate 2 points as 2 separate points. That is known as the resolving power. So if there are 2 points, or let's suppose 2 lines present at a distance of 0.2 millimeters from each other, then they would be differentiated as 2 separate points or lines by the human eye. But lesser than this, if the distance is lesser than this, then they would not be appreciated by the human eye. So if we want to visualize structures which are smaller, tinier structures, then we use the help of the instruments, which are the microscopes. Now, there are many different types of microscopes, but the basic one is the light microscope. And maybe you have already seen different cells under the light microscope in your biology classes. Well, it gives us a magnification of about 1,000 to 1,500 times. This means that the image is the original structure is enlarged by 1,000 to 1,500 times. And the resolving power that we get with the light microscope is now about 0.2 microns. So you can see from 0.2 millimeter, the resolving power has now become 0.2 microns. And this is enabling us to see tinier structures which were not visible with the human eye. So we can look at cells through a light microscope. Then if we want to look at the ultrastructure, that is the structures which are present within a cell in much higher detail, better detail, then we use the electron microscope, which instead of light uses an electron beam. And here the magnification has increased to about hundreds of thousands of times, whereas the resolving power has now become about 1 to 3 nanometers. So here we can observe the structures which are present within a cell, within a single cell. We can look at the organelles. We can also look at the further details, the structures within the organelles with the help of the electron microscope. So whenever you will be looking at the different structures, you should be able to differentiate between a picture which is representing the light microscope view and a picture which is representing the view through the electron microscope. So here on the left, if you follow the laser pointer, you can see this is the blood film, and you can see these red blood cells, and you can see the neutrophils, and you can see all of this through the light microscope, and different stains are used to give us a colored picture. On the right side, you see the electron microscope picture, which is known as electron micrograph. Here you have a black and white picture, which is representing areas which are dark and lighter gray to white. The dark black areas are known as the electron dense areas, whereas the lighter ones are known as the electron lucent areas. So here you can see that a single cell, its structure in much further detail is being visualized, is being focused on with the help of the electron microscope. Okay, so if we are talking about cells, we know that cells are basically of two types, the prokaryotic cells and the eukaryotic cells. Now, since we are going to be talking about the multicellular organisms, we are going to discuss the eukaryotic cells. This is a cell which is typically more than 10 micron in size, and it can be further divided into two major compartments, that is the cytoplasm and the nucleus. The cytoplasm is the part of the cell located outside the nucleus. This is the part which has the organelles, which literally means little organs, and inclusions present in an aqueous gel known as the cytoplasmic matrix or cytosol. Now, these little organs or organelles are the structures that are metabolically active. Now, I want you to remember this important point over here, that the organelles are those structures present within a cell that are metabolically active. So they are further classified into two groups, those which themselves are bound by a membrane, known as the membranous organelles, or those which are not bound by a membrane, that is the non-membranous organelles. So these organelles, if we want to list the membranous organelles, the very first one would be the cell membrane. So here comes the point that I mentioned in the first slide. I told you that the plasma membrane, also known as the cell membrane, which is the outer boundary wall of the cell, is also known as a membranous organelle. The reason the cell membrane can also be called an organelle is because it is not just a simple boundary wall surrounding the cell, but it has many functions. It is metabolically active. It is a dynamic structure. And for this reason, it is considered to be itself an organelle. Then when we look at the further structures within the cytoplasm of the cell, we have other membranous organelles such as the endoplasmic reticulum, which can be the rough, that is the one with the ribosomes, or the smooth endoplasmic reticulum, which is without the ribosomes. Also, there is the Golgi apparatus, which is the packaging factory of the cell, and then come the secretory granules, the transport vesicles. Also are present the lysosomes, the mitochondria. The mitochondria, you know, are the powerhouse of the cells. There are peroxisomes. And then, of course, the nucleus itself is membrane-bound as well, so it is also a membranous organelle. What about the non-membranous organelles? Now, since we are saying that these are organelles, keep in mind they are metabolically active, but they are just not having a membrane. These are the ribosomes, proteasomes, the cytoskeleton. Now, this is the skeleton of the cell, which is made up of structures known as microtubules, actin, and intermediate filaments. Also, the centrioles and the basal bodies are included in this list. Now let's talk about those structures present within the cell that are not metabolically active. So those structures are called cytoplasmic inclusions, and they only contain the products of metabolic activity. These can be pigment granules, such as the lipofuscin wear and tear pigment present often within cells, or they can be the fat droplets. They can also be glycogen granules. And what about the matrix of the cytoplasm, also known as the ground substance or the cytosol? Keep this in mind. This is not merely a filler within the cytoplasm, but it has some important constituents, that is the inorganic ions and the organic molecules. So this is the area, the cytosol is the site of physiological processes that are essential, that are fundamental to the cell's existence. And it is made up of a microtrabecular lattice. Now coming to our primary topic of discussion, which is the plasma membrane. We are going to be talking about the cell membrane, also known as the plasma membrane, and we are discussing its histology. It is also known as the plasma lemma, and as I discussed before, this is a membranous organelle. Now, when we talk about the structure of the plasma membrane in a eukaryotic cell, this is the word that you have to keep in mind, lipid bilayer. The plasma membrane is a lipid bilayer. It forms the cell boundary, and it also forms the boundaries of many organelles within the cell. Now, this is an important point to remember and understand. The plasma membrane is present around the cell as a whole. In addition to that, the plasma membrane is also present around the membranous organelles within the cell. If you recall, I just showed you the list of the membranous organelles. They were the endoplasmic reticulum, the Golgi apparatus, the lysosomes. So all of these membranous organelles, they have their own plasma membrane, and the plasma membrane is a lipid bilayer. And it is a dynamic structure that actively participates in many physiological and biochemical activities that are necessary for cell function and survival. So can we summarize the functions of plasma membrane? Of course, it is a physical barrier, but it has selective permeability, meaning some substances can cross through it while others cannot. And it also provides an electrochemical gradient. Of course, there is a communication of the intracellular and extracellular environment across the plasma membrane. The total thickness of plasma membrane is in 7.5 to 10 nanometers. So since the unit is in nanometers, as I told you, the resolving power of electron microscope is about 1 to 3 nanometers. So it is the electron microscope which will help us visualize the plasma membrane. Although in light microscopy, the plasma membrane can be recognized as a faint line with the help of the extracellular material which is present on the adjacent surface of the plasma membrane. So here is now a micrograph which has been taken through the light microscope, and you can see this tissue slide is showing us these epithelial cells. So this is a slide which is showing us the simple columnar epithelium. These cells are columnar and they're lining the excretory duct of the pancreas. And the stain which has been used is the hematoxylin and eosine stain. This stain gives a pinkish color to the cytoplasm of the cells and a purplish blue color to the nucleus within the cells. Now here on the surface of these cells, on the apical surface, you can see the faint, slightly dark pink line. This is representing the plasma membrane. This is how it is visible through a light microscope. But if we were to look at it through the transmission electron micrograph, then we would see that it is actually consisting of these three layers. Now, these are the three layers which are visible in the electron micrograph. And when we look at these three layers, we understand that this is a trilaminar appearance after fixation with the special fixative, which is the osmium tetroxide. Osmium binds the polar heads of the phospholipids and the oligosaccharide chains. We'll be talking about them further when we're talking about the plasma membrane structure in detail. Well, osmium binds the polar heads on either side of this membrane, the polar heads, and that gives us the electron dense, or you could say the two black lines on the outer and inner aspects of the plasma membrane. And within the middle, sandwiched is the electron lucent part, or the light grayish white part, which is the part which is free of the osmium. These are the fatty acids. So this gives us three layers, two outer black lines and a middle white line. This trilaminar structure is known as unit membrane. And this is what is visible when we are looking at the plasma membrane through the electron microscope. But since, there have been many advancements in science, and when we come to the molecular structure of the plasma membrane, we found out that the plasma membrane is consisting of the molecules. And what are those molecules? We'll be talking about that. But first, let's talk about the appearance of those molecules. They appear to be in the form of a mosaic. Now, what is a mosaic? In very simple words, I've shown this picture to you. Just in very simple words, a mosaic is a tile pattern in which different pieces or blocks, they are brought together to produce a bigger pattern or display. This is known as a mosaic. Now, why is the cell membrane or plasma membrane called a mosaic? Well, because it has molecules which have been arranged and modified in such a way that it gives the appearance of a mosaic. And this is known as the fluid mosaic model. And why is it called a fluid model? Because different advancements in research have shown us that the molecules which are making up the plasma membrane are not static, but they tend to move and shift in their position because the plasma membrane is a dynamic structure. So this is why it is known as the fluid mosaic model. And according to this model, the plasma membrane primarily consists of three components. Now here we're coming to the actual components of the plasma membrane. Remember, there are three components, which are number one, phospholipids, number two, the cholesterol, and number three, proteins. These are the three primary components of the plasma membrane. First, we'll talk about the phospholipids. Now, phospholipids are amphipathic. The phospholipids are amphipathic. This means that they consist of two non-polar long-chain fatty acids linked to a charged polar head that bears a phosphate group. So on one side, we have the non-polar part, which is made up of the fatty acid chains. And when we say non-polar, we mean this is the hydrophobic part. It is afraid of water. This is what hydrophobic literally means. So it is going to be positioned away from the extracellular or the intracellular environment. And on the other side, we have the polar head of the phospholipid, Polar head is hydrophilic, which literally means it is the water-loving head. So this is the part which is going to be facing the extracellular and the intracellular environments. So when the phospholipid has these two parts, the polar group and the non-polar chains, this is what makes it amphipathic. Now let's come to the second component, which is the cholesterol. Cholesterol, you can see over here. Now, this is a simple diagrammatic representation of the plasma membrane made up of the molecules, as I just explained to you. You can see these are the phospholipids. And as I told you initially, this is a lipid bilayer. When we say bilayer, we mean there are two layers of phospholipids. Here is the layer above, and here is the layer below. So these are two layers of phospholipids. That's why it's known as the lipid bilayer. If we were looking at this plasma membrane through the electron microscope, as I showed you before, that would have shown us a trilaminar structure known as the unit membrane. But when we looked at it in further detail at the molecular constituents of plasma membrane, we see that it is not really consisting of three layers, but in fact, based on the constituents, it is consisting of two layers. These are 1 and 2, the two lipid layers. But they appeared as trilaminar in the electron micrograph because these phospholipid polar heads, which you can see on the upper and lower aspects, they took up the osmium fixative and they gave an electron dense appearance, which is the dark band 1 and 2, these two dark bands. And the middle portion, which consisted of the non-polar fatty acid chains, they did not take up the osmium stain, the osmium fixative. So that is why this area appeared lighter. That was the electron lucent area. And this gave the appearance of 1, 2, and 3 layers in the electron micrograph. But keep this in mind, whenever you talk about the fluid mosaic model and when you look at the actual constituents, the plasma membrane is a lipid bilayer. One over here and the second over here. And the important thing to notice is that the polar heads are present externally and internally facing each other are the non-polar fatty acid chains. And here I told you is the second constituent. You can see the cholesterol. It is present within this plasma membrane. So these parts which have the polar heads are known as the hydrophilic surfaces, and the part in the middle is known as the hydrophobic region. Also keep in mind on the outer aspect of the plasma membrane, you can see the sugar chains which are attached to the phospholipids. And this arrow you can see over here points towards this cell coat or glycocalyx, because eventually when you look over here, you can see over here in this electron micrograph, this is the glycocalyx attached to the outer aspect of the plasma membrane. This glycocalyx consists of the sugar chains which are attached to it, over here shown to us in green. So the second constituent, cholesterol, is a sterol lipid, and it is inserted at varying densities amongst the phospholipids. It has a very special function. It restricts the movement and it modulates the fluidity of all membrane components. So even though the cell membrane is a fluid structure, yet there is some restriction to the movement of the structures within the plasma membrane. And this restriction is a function of the cholesterol within the plasma membrane. Now, the third constituent are the proteins. There are lots of proteins present within plasma membranes, so many that they are making up about 50% by weight of the plasma membrane. And these proteins can either be embedded within the lipid bilayer. In that case, they are called the integral proteins or transmembrane proteins, or they might be bound to the other proteins which are present within the lipid bilayer. In that case, they are known as the peripheral proteins, or they might be multi-pass proteins. When the integral proteins pass through the plasma membrane several times, they are known as the multi-pass proteins. So here we can see the fluid mosaic model. So here we're looking at the fluid mosaic model of the plasma membrane. And let's look over here at this diagram. We can see that this is a lipid bilayer, which means there are two layers of the phospholipids, and the fatty acid tails are the non-polar tails which are facing each other, whereas these purple balls are representing the polar heads, which are the hydrophilic parts of the phospholipids. Also, we can see these proteins which are present within this plasma membrane. Here is an integral protein because it is embedded within the plasma membrane. Over here, let's look at these peripheral proteins, which are not directly embedded within the plasma membrane, but they're attached to the integral or transmembrane proteins. Also, we can see on the outer aspect of the plasma membrane, this is the part which is facing the extracellular compartment. Here are the sugar chains which are attached, and as I mentioned before, they will form the glycocalyx on the external aspect of plasma membrane. And here, this diagram B is simply showing us that the parts of the plasma membrane have been split open along the fatty acid tails. And here you can see this is the two-layered or the lipid bilayer. So if we do a simple experiment to check for the fluidity of the plasma membrane, then this is the experiment that was done for it, in which two types of cells were grown in tissue cultures. You can see here on the right is the cell which has the fluorescently labeled transmembrane proteins in the plasma lemma. Whereas on the left, you can see this cell, its plasma membrane has not been labeled. Now, when these two cells were brought together and fused together into a hybrid cell, then we followed the fluorescently labeled transmembrane proteins, and we saw that they had covered the entire surface of the new hybrid cell. This shows that the plasma membrane is not a static structure, but the proteins and the constituents within this plasma membrane, they are moving, and they're not static. So we should also keep in mind that these movements do have some restrictions. And the recent evidence in research has shown that the movements of proteins in the plasma membrane is not so random, but it is restricted. Now, what are the factors that restrict the movements of the proteins in plasma membrane or the fluidity of plasma membrane? Well, we should keep in mind cholesterol. Cholesterol, as I mentioned before, is the constituent responsible for restricting the lateral movements of plasma membrane. And it is present in the form of lipid rafts. These are microdomains of plasma membrane that control the movement and distribution of proteins within plasma membrane. The lipid rafts have a high concentration of the cholesterol glycosphingolipids. So remember, cholesterol is the glue which holds these rafts together. And there are two types of lipid rafts. They can be planar or they can be caveolar. Now, this is a fluid mosaic model of plasma membrane where I want to show you the lipid rafts responsible for restricting the movements of the proteins within the plasma membrane or restricting the fluidity of the plasma membrane. Here you can see is a lipid raft in front of you, and it is consisting of the cholesterol glycosphingolipids. Here are the cholesterol molecules present over here as well. You can see them. Also, these green branches sticking out on the external aspect of plasma membrane are representing the sugar moieties, that is the carbohydrate chains, which form the glycocalyx. Then here are the embedded proteins, which are integral or transmembrane proteins, and attached to them on the outer aspect or the inner aspect of the plasma membrane, but not directly embedded within plasma membrane, are these peripheral proteins. So the factors restricting the fluidity of plasma membrane are also the cytoskeletal attachments of the proteins with the of the plasma membrane. So you can remember that cytoskeleton was the skeleton of the cell which is present within its cytoplasm. So it is anchoring lots of parts of the plasma membrane, restricting its fluidity. Another factor is tight junctions between epithelial cells. They also restrict the lateral diffusion of the unattached transmembrane proteins and outer layer lipids. Then there's another factor known as scaffold proteins, which maintain spatial relationships between enzymes and signaling proteins. So these are the multiple factors other than cholesterol which are responsible for restricting the fluidity of the plasma membrane. Also, if we look at the proteins within the plasma membrane, they have multiple functions. They can serve as channels for the transport of molecules, and they can also serve as carriers or pumps that use energy for transporting substances against a concentration gradient. And as I told you before, that the proteins within the plasma membrane, if they are integral, they can be one pass or they can be multi-pass. One pass is when it passes through the plasma membrane only once, but if it passes through the plasma membrane multiple times, it is known as a multi-pass integral protein. Here are the different mechanisms by which the substances can be transported across the plasma membrane. So here in diagram A, you are looking at phagocytosis. Now, what happens in phagocytosis is that the plasma membrane, it throws out these projections or finger-like extensions known as pseudopodia for engulfing or taking in bacteria or other particles and internalizing it into a membrane-bound structure which is known as a vacuole. Also, it is called phagosome. Let's look at diagram B, which is penocytosis. In this case, the plasma membrane, it invaginates inwards, or you could say pinches inwards to form a tiny vesicle which contains a drop of fluid with certain substances. And this is known as a penocytotic vesicle. Here you can see in diagram C is endocytosis, that is receptor mediated. So endocytosis, which is receptor mediated, is also a form of the transfer of substances or their intake by the plasma membrane, in which it forms a cytoplasmic vesicle. But this is happening with the help of receptors. Receptors are the proteins you can see present in the plasma membrane that bind the ligands and bring them inside in the form of a cytoplasmic vesicle, also known as an endosome. So these are the multiple ways in which the plasma membrane is performing these functions. So now that we have talked about the plasma membrane, we have discussed the basic structure and also the functions. Let's look at this MCQ over here. In transmission electron micrograph preparations of cells, the cell membrane often appears as a trilaminar structure having two parallel dark staining components on either side of an unstained middle layer. This central poorly stained region of the membrane is primarily responsible for which of the following functions? Now, if you recall, I told you that the trilaminar appearance, which is known as the unit membrane appearance of plasma membrane in the electron micrographs, it is showing us the middle part, the sandwiched part, which is the electron-lucent part. It is representing the fatty acid chains of the phospholipids. And this is the non-polar part. And since this is the non-polar part, here you can see that it is non-polar, which means it is hydrophobic. And since it is hydrophobic, it creates a barrier to the water-soluble molecules. And this is why D is the correct answer for this MCQ. Regarding the plasma membrane, self-recognition, attachment to other cells, and extracellular matrix components is a function of, well, all of these functions are due to the glycocalyx code. So this is what you have to keep in mind, is the function of the sugar moieties, which are attached on the outer aspect of the plasma membrane. Regarding proteins in the plasma membrane, the following statement is true. Now, if we look at all of the MCQs, since we're selecting the true one, we have to see if the others, we have to start from the very beginning. Let's look at option A. All integral proteins are bound rigidly in the cell membrane. We know this cannot be true since we discussed that the proteins, they are having some, although restricted, but they do have a certain amount of lateral movement within the plasma membrane. So they're not bound rigidly in the cell membrane. So A cannot be the answer. Let's look at option B, which says all integral proteins pass through the cell membrane several times. I told you that the integral proteins can be either one pass or multipass. If they're passing through the cell membrane several times, it means they're multipass. But not all of them are multipass. They can also be one pass in which they pass through the plasma membrane only once. So this also cannot be the correct answer for this MCQ. Proteins form 5% weight of the cell membrane. We know it's not 5%, it is 50%. So this is also not the correct one. The correct answer is option D, which says that some integral proteins protrude from the inner surface of the cell membrane. Well, that's true. There's nothing wrong about that. So this is the correct answer for this MCQ. Okay, so this concludes with today's discussion on the plasma membrane. And I hope this was interesting for you, and you got the important concept related to the appearance of plasma membrane in a light microscope, then the appearance under the electron microscope, and then, of course, the molecular constituents, which is the fluid mosaic model. And I have also included a short description of the plasma membrane with the help of drawing on a schematic diagram in which I have explained all of these constituents by drawing them out for you. So you would remember that plasma membrane basically consists of three components, that is phospholipids, proteins, and cholesterol. So if you like today's discussion, don't forget to like and subscribe to my YouTube channel, AnatomyMentor. You can do that by simply clicking on the picture, on my picture, which is going to appear at the end of this video. And also don't forget to hit the bell icon so you can be immediately notified of further uploads, whatever presentation I have to share with you, so you will be getting it immediately. Thank you, take care, enjoy your studying, and stay blessed.